Welcome to today's ASHI Educational Initiatives Committee webinar. We are excited to have you join us for this informative session designed to enhance your knowledge and expertise in the field. My name is Ahmed Mustafa, and I'm the Vice Chair of the ASHI Educational Initiative Committee, and I will be your moderator for today's session. We have just like a few housekeeping items. This webinar is recorded and will be available on the ASHI U for future reference. You will receive an email by the end of the week with a link to complete an evaluation. Upon completion, a continuous education certificate will be emailed to you. You can ask any question you want through the chat function. Now it's my pleasure to introduce our dear friend and colleague, Dr. Karen Sherwood, also known as the Nanopore celebrity. Dr. Sherwood is a clinical associate professor at the University of British Columbia and also the HLA lab director at Vancouver General Hospital, where her research focuses on precision medicine and genome technologies in transplantation to enhance patient outcome and reduce rejection risk. Today, Karen will give us a talk titled High Resolution Sequence in HLA Laboratories, Past, Present, and Future. The floor is all yours, Karen.
Thanks, Ahmed. That's a very generous introduction. So yes, I would like to thank the organizers for inviting me to speak at this ASHI educational webinar. And my first slide is just no conflict of interest, and I'd like to also acknowledge that the work and research that I am part of is carried out on land that is on the traditional, ancestral, and unceded territory of the Musqueam people. So as Ahmed has mentioned, I'm hoping that there'll be 3 objectives that you guys will have a little bit more information about by the end of my talk. I will first just briefly go over the historical developments in sequencing, specifically to clinical HLA, and then how these advances have impacted most of our work in the clinical practice, and then a little bit about some emerging trends and the future impact of how those trends might change the way that we do our transplant immunology. So just a short journey through the history of DNA sequencing. I'm sure we're all familiar with, I don't know if you can see my pointer, but from the far left, familiar with Gregor Mendel's work with peas in the 1860s, and then jump forward to 1869, Frederick Mesher isolated what we now call the DNA, the molecule that's now called DNA. And in 1953, Watson and Crick revealed its beautiful double helix structure. But the real revolution in sequencing came far later. It started really in the 1970s, and that's when researchers developed the first methods to actually read DNA sequences. Now, it started really with the Sanger sequencing, which many of you may be familiar with or have heard of, which gave us the ability to sequence one gene at a time with remarkable accuracy. And from there, the pace accelerated dramatically. Pyro sequencing and other next generation approaches allows us to sequence millions of fragments in parallel, which is different from Sanger sequencing, and which made the Human Genome Project possible. The Human Genome Project was a landmark achievement in 2001 and showed us that the whole genome is not just theoretically feasible, but practically achievable. And I'll talk a little bit more about what that looks like moving into 2026. And today's long reads are even more affordable than the next generation sequencing methods, and these are a direct descendant of this whole history. So what we now refer to as next generation sequencing, also sometimes called 2G or second generation, is really the product of several distinct technological eras in DNA sequencing. So at first, it starts with first generation Sanger-based methods, where we could read one fragment at a time with very high accuracy, but it's exceedingly low throughput and high cost. The OG 454s from Roche, I don't know if many of you remember those. Those were the ones that used the clonal amplification by emulsion PCR, that's the square in the middle, and they created clonal beads for sequencing. Then we had the ion torrents, originally from Life Technologies. Many of you may have actually used these in the HLA labs. Initially, I know I had a couple of ion torrents when they were actually part of the PGM suite of technologies. That is a proton detection-based sequencing method. Life Technologies also had the SOLID platform, SOLID being sequencing by oligonucleotide ligation and detection platform, and that worked by ligating fluorescently labeled probes to a DNA template. That's slightly different from what is really now considered the gold standard of short reads or second generation sequencing, which is the one on the far right. That's Illumina's proprietary bridge PCR method, which then uses those clusters to use fluorescently labeled nucleotides to build new strands. This is just a brief overview of long read or third generation technologies. There are really two key approaches, main flavors of third generation, sometimes also called single molecule sequencing. And the first is a polymerase-based system that allows you to watch a single DNA polymerase copy one DNA molecule in real time, base by base. This approach allows us to get very long reads and avoids many of the amplification biases of short read platforms. And the other approach is the nanopore system, where the DNA strand itself passes through a tiny pore and the sequencer reads the changes in electrical current to infer the sequence. There's no synthesis step in that approach, and the reads can also be extremely long, and the data stream that comes off the instrument comes off in real time. And these two approaches, which are really the leaders in the third generation sequencing field, these two approaches are what make it possible to think about full-length phased HLA genes and sequencing the complex immunogenetic regions as part of routine standard of care. So the evolution of HLA typing methods, many of you might remember or have learned about HLA phenotyping back in the 1970s, all the way through to when HLA, the field of HLA started to look into next generation sequencing in the early 2014s, 2015s. So sequencing in clinical HLA has evolved from Sanger-based exon-only sequencing for a limited number of loci to today's short and long read approaches that can deliver full gene, phased, high resolution genotypes across the whole HLA region. So each technological step, either PCR-based SSO, SSP, which I'm sure many of us still use, Sanger sequencing-based typing, which we have used in the past but has now been phased out, next generation sequencing also, which we've used and is now phased out, and to third generation long read sequencing. All of these steps have allowed us to reduce ambiguity, has increased our ability to get high resolution and expanded clinical impact. So this is just a table breaking down the different eras that many of you might have been familiar with. I won't read through each section. I'm happy to share my slides after as well if anyone is interested, but I'll just briefly go through the early molecular HLA phase with SSO, SSP, and SBT. These interrogated limited exons and often left unresolved phasing and multiple possible allele combinations, especially in the highly polymorphic loci. We're all familiar with this over the years pre-sequencing. So when we move into short read NGS panels on platforms such as Illumina or Ion Torrent, these now routinely sequence the entire HLA genes, including introns and untranslated regions. And this substantially reduces ambiguity and improves concordance with reference results. High coverage, full gene, and now long read technologies have definitely increased the accuracy, and now we get very high concordance with all of the HLA variants that are in the database, and this is to a single base resolution. And as the field moves to long read sequencing as standard, ambiguity reduction will continue, as these methods are particularly valuable for structurally complex loci and for samples where short read methods leave residual ambiguities. Turnaround time and workflow integration has also changed rapidly between the era of labor-intensive Sanger workflows to the now comprehensive, rapid, high-resolution genotyping using long-read platforms. So this is probably a throwback for many of you. I will admit that I didn't actually have to use any of these trays, but the pre-sequencing and early molecular era clinical HLA typing began with serological assays in the 1950s and 60s, using complement-dependent cytotoxicity and later flow-based methods to define antigens at a broad level. These approaches depended on collecting viable lymphocytes and available antisera, which was messy and non-standardized, and they gave limited resolution and could not distinguish many of the alleles now recognized within a given serological specificity. With the introduction of PCR into the clinical space in the mid-1980s, this enabled DNA-based typing with sequence-specific oligonucleotides and sequence-specific primers, SSO and SSP assays. These were initially focused on polymorphic exons of class 2 and then class 1 genes. These methods did improve accuracy and did allow genotypic nomenclature, but still left frequent ambiguities because only selected codons were interrogated and cis-trans phase remained unresolved. So then we moved into the Sanger sequencing-based era, which was introduced in the 1990s and was first routinely used for DNA sequencing in clinical HLA around then, and typically targeted exon 2 and 3 for class 1 and exon 2 for class 2. By directly reading nucleotide sequences, SBT significantly improved allele level resolution compared with probe or primer-based methods, and eventually became the gold standard for many hematopoietic stem cell transplant programs. However, SBT is labor-intensive, it's relatively low throughput, and did produce phase ambiguities when heterozygous positions overlapped within amplicons, leading to multiple possible allele combinations without additional cloning or group-specific amplification. So this led to limited comprehensive multi-loci high-resolution typing in time-sensitive clinical workflows, and especially in large donor registries. So from there, we moved into NGS era, next-generation sequencing or sequencing by synthesis. Many of your labs may already be doing this or currently be doing this, where we get single base resolution, massively parallel multiple samples with a much lower turnaround time than Sanger sequencing. And because you can batch multiple samples into a single run, which fits with a much required time-sensitive clinical turnaround times. So then we move into, or start to move into, third generation long read sequencing. So more recently, third generation platforms such as PacBio, which recently came out with this hot pink version, which I quite like, and Oxford Nanopore have enabled sequencing of long amplicons or captured regions, spanning entire HLA genes, and in some formats, multi-gene haplotypes. Single molecule long read data directly resolves phasing across distant polymorphisms and structural variants, and it can address residual ambiguities left by short read methods and clarifying complex alleles. Clinical reports now describe the implementation of long read HLA typing for high throughput, full gene phasing with rapid workflows with real-time analysis, and can integrate with these with other genomic assays, marking the latest milestone in the evolution of sequencing for HLA. These advances complete a historical arc from phenotypic serology to comprehensive sequence-based haplotype-resolved genotyping that underpins contemporary transplantation and precision immunogenetics. So then we move into how these technological advances have enhanced the accuracy and application of HLA sequencing in clinical practice. So here, this is a paper from Robinson. It was published last year, 2024, the 25-year outcome of the IMGT database. And the top graph here shows how the sequence coverage has changed over the lifetime of the 25 history and has been impacted by emerging technologies, which have allowed for the submission of both coding and full-length genomic sequencing. Initial releases of the database were focused on exon 2 and 3 sequences of class 1 and exon 2 of class 2 alleles. But as full-length coding sequences became more easily sequenced, the database expanded rapidly and it started to include these Later, the full-length genomic sequences of HLA class 1, as it became routine as part of HLA labs, and these sequences were added to the database, it's now possible to have very limited challenges due to sequence length, and the increased number of homopolymers and repeat regions in the intronic regions no longer poses an issue. And the bottom graph here also shows that the growth of new alleles has traditionally been assumed to be down to the fact that there are many intronic variants of these HLA alleles. But actually, the team were able to show that the categorization for each allele, there were three main periods which differentiated this increase in submission to the database. The first was primarily probe-based methods, as we mentioned before, and those were the ones that identified variants within the ARD. And then once we sequenced the ARD, this enabled us to identify silent variants. And finally, next generation sequencing and now long read sequencing, it's extending that beyond the ARD, allowing identification of intronic and UTR variants. So as these technological advances in short and long read sequencing have now enabled us to do full gene phase resolved typing with fewer ambiguities and fast turnaround time and broader clinical application, these legacy serologic and PCR-based methods may eventually become a thing of the past. And the clinical impact for these is that the shift from serological to DNA-based typing, particularly single base sequencing, high throughput sequencing such as NGS, has fundamentally transformed clinical practice in transplantation medicine. For stem cell transplant, we know that this enhanced resolution of DNA sequencing allows for more precise matching of donors and recipients, which is crucial for reducing complications like graft versus host and graft rejection in stem cell transplant. We also know that the faster turnaround time with modern sequencing means that we can find matches faster and process transplant faster, which is critical for transplant survival. Having high resolution also allows us to increase the donor pool. The ability to type to a high resolution has expanded the effective donor pool, especially for ethnic minorities who are less represented in traditional registries. In addition, we know that high resolution typing is actually required for the definition of epitopes. I'm sure you've probably seen me present this slide before, where we go from HLA alleles, sorry, Paul's just walked in and seen my friend, to now being able to match based on epitopes to reduce immunogenicity in kidney transplantation. And we'll actually be rolling that out in a month, in January 2026. And of course, with disease association studies with many of our labs also do these enable better understanding of the HLA associations with autoimmune and diseases, and allows us to personalize treatment strategies. I'm just going to skip this. That was just that slide. Okay. So international HLA and immunogenetic workshops have been running for a long time. I'm sure many of you have been to some of these. And they provide much of the infrastructure and the reference material and consensus needed to move the HLA sequencing from experimental use into routine clinical practice. And they continue to shape how emerging sequencing approaches will be standardized and interpreted. The first histocompatibility testing workshop was organized by Dr. Amos and held in Duke University in 1964. That's the top of that. And there were only 23 investigators at that workshop, but they were engaged in defining what was recently discovered, the human leukocyte antigens. But it wasn't until 1967 workshop that the standardized nomenclature of naming the HLA genes began. Between then and 1987, the international workshops were primarily organized to define the HLA genes. But it wasn't until the 10th histocompatibility workshop in 1987 in New York that a DNA typing method was established for typing HLA. That was actually using RFLP7 plots. The earliest workshops established a template for sharing common cell panels, reagents, and analytical frameworks, which later made it possible to compare and validate new molecular methods, including next-generation sequencing. And all subsequent international HLA workshops, although now have expanded to tackle other areas, still have a component for providing increasingly sophisticated documentation of the extraordinary diversity of the HLA complex. So then the 17th International Workshop, they explicitly focused on NGS for histocompatibility immunogenetics, organizing a pilot efficiency testing program in which multiple laboratories sequenced standardized cell lines using different NGS platforms. And the consensus genotypes from these multi-center efforts became benchmark data sets highlighted in publications from IMGT and repositories, and these validated clinical pipelines. And then the present workshop, the 18th workshop, these will actually emphasize using next generation sequencing and long read sequencing for full length and hemizygous sequencing of HLA genes, including non-coding regions, as well as promoter and extended haplotypes, haplotype characterizations for loci such as DP. And these efforts in these workshops and in future workshops generate curated reference sequencing and haplotypes that underpin imputation panels, epitope mapping, and the interpretation of variants detected by clinical assays. So now I will go very briefly into some of the emerging advances in the sequencing space that may be coming to an HLA lab near you. Roche were awarded, along with the Broad Clinical Labs in Boston Children's Hospital, they were awarded the world record for the fastest sequencing of a human genome using their newest sequencing by expansion, SBX technology. So it is recorded as sequencing an entire human genome in 4 hours. I will just highlight that that's 4 hours of sequencing time. With highly specific NVIDIA compute capabilities, and that comes relevant later on when I talk a little bit more about the emerging technologies. But they were able to report off these results within 24 hours of the sample draw. Illumina also has a few new tricks up their sleeve, including simultaneous methylation profiling. And in general, I think we'll start to hear about short and long read hybrid methods where you combine both short and long reads. That will become a much more popular implementation of in clinical labs. And of course, integration of AI into anything and everything we can get it into. So I'm hoping this video will work. It's just a very short video to SBX. I don't know if many of you are familiar with sequence by expansion. It's an entirely new paradigm in nucleotide recognition. So I'll just play this. I hope you can hear it. So I hope you guys were able to hear that. I didn't test that before. So I'll move to the next slide, just 1 second. So that's sequencing by expansion, and this was the technology that was used to produce the fastest sequenced human genome in 4 hours. And this is just a figure from that paper. It was published in October in New England Journal, and you can see that they actually had the breakdown of when the sample came in, how much time it took to do the sequencing, and how long it took to report out on that genome. So it is within 24 hours, and actually the expandomer synthesis is only about an hour and a half, with sequencing only being 20 minutes. So it is exceedingly fast. This is a whole human genome. It is still very, very new and is not currently commercially available. As I mentioned, there are other emerging trends in sequencing. So these are some trends in the short read space. Illumina has come out with a new chemistry that will allow for simultaneous sequencing, DNA sequencing and methylation. It converts 5-methylcytosine to thymine in a single non-damaging step, and allows for simultaneous detection of both the standard DNA bases and the 5th base of DNA, the 5-methylcytosine. Illumina is also releasing something called constellation sequencing. So this combines short reads that also provides long-range genomic information. It's not long reads, it's long-range information. So essentially they're doing the sequencing, but in 2 sort of wells, and somehow they're connected. Again, this is not released until 2026, and so there's not a lot of information. It also does require its highest capacity instrument, the NovaSeq, which I'm not sure how many labs have. Usually these are used in registries, sort of high throughput registries. And there's a new sequencer on the block, the Biosciences Aviti. This is a benchtop design, but has surpassed the Q40 level accuracy on 300 base pair reads. So that's, outperforming even Illumina gold standard Q levels of accuracy. Some emerging trends in long read sequencing. There's going to be improved accuracy. I know that people still discuss the accuracy of some of the long reads. I don't think that's going to be much of a problem with many of the applications that we're intending on using right now. This image on the far right, the 2 black boxes, that's actually Roche's Revio system and their Ontocom or something. It's both the long read is the big one and the short read is the small one, and you can actually combine them and produce these combined hybrid approaches, which I mentioned before, long read with short read for increased accuracy, but with the benefit of having long reads to span large pieces of the genome. And obviously, automation is another thing that many of these companies will be trying to bring into the field that will assist with clinical adoption. A lot of these long read sequencing companies have been trying to move into the clinical space. Short reads have been there for a while, and so clinical adoption is something that many of these companies are starting to think about. So while there is a lot of excitement about advances from 2G to 3G to 4G sequencing technologies, the applications of these in the HLA clinical practice really comes down to the clinical utility of the data that these technologies afford us. And so I will cover a little bit about that in some later slides. I'll just finish going over some of the other emerging trends in 2026. So specifically for nanopore-based sequencing, this isn't possible with smart sequencing. As far as I am aware, I don't know everything in this space, but I try to keep up as best I can. Nanopore-based sequencing has officially, as of May 2025, moved into proteomic sequencing. So proteomic sequencing is highly complex because with proteins, there are more than 400 post-translational modifications that can occur. It's not like 4 bases or DNA modifications where there's only a couple of options. This is a much more complex signal. The squiggles will become much more complex to read. So Oxford Nanopore laid out in their annual conference a roadmap to move from DNA, RNA, which they currently do very well, into direct protein analysis, starting with a peptide-based detection workflow, and then progressing towards full-length protein identification on the same Nanopore platform, with their stated goal is a modular multi-omic system, and where you can just change the pore chemistry and allow the same device to sequence DNA, RNA, or proteins. So it's the same thing. They either sequence these peptides, and they've actually started producing some early release data. You can see that the squiggles, which you guys might be familiar with, actually have these very familiar profiles where they have the peptides that come through, and with each end being a DNA sort of barcode of that peptide. Again, very, very early days, but keep an eye out for some of these in 2026. And of course, AI. The emergence of NGS, together with the necessity of technological advances, drives our increasing ability to uncover complex and previously hidden genetic aberrations. However, our current capacity to interpret their functional and clinical impacts remains constrained, and this is where AI enters the room. And while I'm less keen on having AI thrust at me at every opportunity, I'm pretty sure I can't even buy my tea anymore without an AI agent popping up. I do think that the multi-omics space is where AI really could be powerful, especially as it becomes more, both the omics space and AI itself becomes more scalable. And we will be seeing the use of AI in the HLA genomic space, if not already, definitely in 2026. And you can actually see that as AI markets increase, the cost of sequencing is coming down. Oh, yeah, so you can see now we're almost at the, actually this is a little bit outdated, but we're down to 300 and something US dollars for a single full human genome. And the we're hoping to hit the $100 genome at some point in 2026. But I don't think it's any coincidence that as the cost of genomes go down, the cost of NVIDIA stock prices go up and up and up. And Paul and I always joke that we should really have bought into NVIDIA when we knew years and years ago, pre-AI and pre-genomics, because many of the genomic platforms use NVIDIA chips for their real-time base pulling. So NVIDIA is just going to be raking in the cash. So all of that to say, why do we care? What are the future impacts of all of these trends, technological trends on transplant immunogenetics? So having high resolution full gene HLA data supports more precise donor selection in stem cell transplants, which I mentioned before, including fine mapping of permissive versus non-permissive mismatches. We know that high resolution HLA typing also allows us to do epitope mapping and therefore match, using those as match strategies. These were not possible before with low resolution typing. And better resolution and phasing allows us to improve risk stratification for graft survival and GVHD, especially when we're using partially matched donors when fully matched grafts are not available. We're also using these rapid sequencing platforms. We're now able to look at non-classical HLA and MHC linked genes, also the CUR genes and other innate receptor genes that might have an impact on transplant outcome. Other variants that affect, for example, the organ vulnerability, such as APOL1. And something that I've been looking in more recently is pharmacogenetic variants in both donors and the recipients. This can modulate the way that we personalize therapeutic management. And also, while we begin to understand there are many different mechanisms that govern expression of HLA proteins, we are as yet unable to predict the amount of HLA protein on the cell surface of any one individual, or even on different cell types within the same individual. So this is where doing RNA-seq, using these rapid platforms, identifying polymorphisms in promoter regions, looking at the tissue or context-specific differential expression of HLA, and alternate splicing of HLA genes really could change the way that we understand how the immune system and the HLA works. Prediction of transplant risk based on the measurement of HLA antibodies alone, without a clear determination of cognate antigen expression on the target cell, for example, in the kidney, like the renal endothelial cells, is an oversimplification of the problem and may deny some patients a viable transplant or expose others to greater risk of antibody-mediated rejection. So having transcript-guided long-read sequencing and expression assays, as these become more accessible, using these long-read platforms, and we can integrate information from isoforms and expression data into donor selection, DSA, risk stratification, and other tolerance strategies. These are coming, but right now are unrealized opportunities that we could move forward in the space of transplantation. As I mentioned, some of the newer platforms will permit simultaneous detection of the DNA, as well as DNA variation and methylation. Now we can include the immune gene promoters, differential expression of the genes, looking at structural variants, including copy number variants. We can identify single nucleotide variants expressed or otherwise, insertion and deletions that alter the expression of the HLA variants, and looking at in RNA differential splice events and allelic imbalance. All of these things are possible actually on a single platform now, which will allow us to broaden our understanding of the immune response. So how are we going? So another future direction could be clinical whole genome sequencing, which, as I just showed you in that milestone paper in October, we can now do in about 24 hours. That is quite a lot of information. And yes, whilst ongoing improvements in long read accuracy and error correction and costs are coming down, and they're expected to make fully phased haplotype resolve profiles, really easily accessible, we still have to understand the underlying complex interactions between all of these things, and integration of these data with structural and epitope level information, as well as AI-driven risk prediction, all these cool tools everyone's been talking about. These will likely broaden the clinical impact of sequence-based HLA typing beyond the current match paradigms. There are also some expanded clinical applications outside of just transplantation, HLA transplantation. So beyond transplantation, sequencing-based HLA genotyping also could underpin individualized cancer vaccine design, autoimmune disease association studies, and as I mentioned before, pharmacogenomics, where allele level resolution is critical for predicting adverse drug reactions. And using long read sequencing and implementing that in our clinical program, we can begin to provide single comprehensive HLA tests that can be requeried over a patient's lifetime for emerging HLA associations and expanding precision medicine applications in our routine care. So that brings me to a little bit of data that we have produced in that space. So as I mentioned, long-read sequencing is now our routine HLA genotyping method. We use a Nanopore-based platform. It looks like whole genome sequencing could be, in the not too distant future, a test that is routinely done for patients. But there is an interim step in that space because there are some limitations still and obstacles that we need to be aware of if we decide to move into the whole genome space. So we've tried this approach called adaptive sampling. It is specific to the Nanopore-based platform. And here, this is just a differentiation of how short reads versus long read sequencing, but using adaptive sampling might allow us to combine and assess multiple regions of interest simultaneously. So if any of you are not familiar, adaptive sampling, sorry, that's a typo, using the Nanopore platform, this process, which was formerly known as read until, selectively sequences DNA strands based on predefined regions of interest, or ROIs, within the genome. And as the DNA strands enters the nanopore, it's initially analyzed, the first 200 to 400 bases are read, and then aligned to your reference genome, human genome. If that strand in the pore matches the target ROI, it will continue to pull the DNA through the pore and continue sequencing. If it does not match a region of interest, the system will eject it by reversing the electrical potential that's within the flow cell, and the DNA strand will then get popped out. And this approach actually optimizes sequencing efficiently by freeing up nanopores to process only the relevant strands within your region of interest. This also takes native DNA, and as amplification is not required, it's long read of native DNA, this retains base modification information that can also be generated at the same time. In addition, using this kind of approach, regions that cannot easily be amplified due to sequence composition, such as high CG content or lots of polymer repeats, these areas can actually repeated, so enriched. So our approach here in Vancouver, most of this work has been done by one of my team, Sarah Gratz-Dieck. We have tried to test a rapid all-in-one assay that achieves up to 100. I think we're actually at more than that, 200 ROI coverage in a 2-day turnaround. And this actually effectively replaces about 10 clinical tests. The data quality that we get is suitable for high confidence, small and structural variant calling, allele phasing, and DNA methylation for profiling in all of the ROIs that we selected in our test bed, as well as simultaneously producing a low pass whole genome in the background. And the reason you get this low pass, sort of 10 to 15x whole genome in the background, as I mentioned, when this approach requires that the pore pulls through the first 200 to 300 bases to check if it actually is part of your ROI. So over time, you get these sort of short, almost a short read, the first 200 to 300 bases in the background before it pops out the fragment. So you get this low pass whole genome as a sort of byproduct. So our assay that we developed includes 164 regions of interest. It includes the complete HLA locus, which is 5.5 megabases, and the T-cell receptor gene locus. We've included 450 ISBT blood group genes, and over 100 of the CPIC non-HLA pharmacogenomic loci for drug response prediction. So this is just an example of some of the sequence enrichments and phasing. So this is a enriched region of interest here. This is the ABO, part of the ABO gene. You can see that this actually spans thousands of kilobases, 15 kilobase here. And I think this goes between 100 and 300x coverage. I also just wanted to show you, I left in the background, this is background whole genome coverage. So you get about 10 to 30x coverage in the background on your region, the region that is not your target enrichment area, as well, with an average read length of 400 bases, which is still, it's useful size that you can obtain. And this also includes the phasing information. The allele 1 and allele 2 are able to be phased by SNP positioning. This is just an example of some of the structural variants that we were able to identify in some of our samples. These are indels that were identified in the A4 GALT gene. This is here, this is an insertion as well in the ABCC4 gene. And this here is an insertion as well in the ABO gene on chromosome 9, a couple of insertions there. This is very early days, so I don't have too much data on the methylation, but as I mentioned, because it's not an amplified assay, you're using native DNA, we're able to, at the same time, identify modified bases, looking at DNA methylation. And this is just one position that we found that showed a different tagged DNA molecule. So that was some of the data from our all-in-one test. We're hoping to get that published before the end of the year. And it really is just an example of how we could have comprehensive risk assessment for immune risk in transplantation as an all-in-one test. This test allows complete sequencing of the HLA genes, which is whole gene and whole region, in fact. In this setting, we can also assess for non-HLA immunogenetic loci, MIC-A and MIC-B. Some of these are coming back in fashion again. These genes are important. They're known to modulate the immune response. We can sequence for many of the PGX genes and identify which one of those are actionable. These inform metabolism and responses to common immunosuppression drugs used in transplantation. And sequencing of the blood gene groups also enables matching beyond routine serology for minor antigens and lowering the risk of transfusion reactions. So I just wanted to mention, as I said, that we've been having, the HLA space has been having HLA workshops for many years. The next one, the 19th International Workshop, is in May 2026 in Japan. I will do a shameless plug. I do have a project on there, ABO histocompatibility, if anyone is interested. I'm happy to share the link with you, where we'll be looking at ABO antibodies and ABO specific, not the minor blood group genes, but ABO specific genotyping in a set of patients. So hopefully see some of you there. There will also be many other projects as part of this workshop. And yeah, so with that, I'd like to end my webinar and say thank you to everyone. It looks like everyone everyone that's on here, but specifically to the members of my HLA lab, BC Immunology lab here at BGH in Vancouver, all of our collaborators at BCH, and all my team members on the Genome Canada Consortium, which includes Ahmed. See Ahmed's come back on. And of course, a recognition for all our patients and their families for their contributions.
Hey, thank you so much, Dr. Sherwood, for this fantastic presentation. I always learn from you something new every time you present. So I'm reminding everyone that all the question can be added to the chat, to the Q&A box. We will try to answer every question, but don't worry if your question is not answered, we will respond to you, or Dr. Sherwood will respond to you by email. So until we get some questions, I have actually a question, and usually come to my mind because now you presented things about long reads, and please, Karin, fix if I'm saying something wrong. So my understanding that Nanopore sequence generate long reads, but many of the HLA software use short read segment for the analysis. Is that correct, or I have misconception in that?
You mean like commercially available software or like?
Yes.
Yeah. So I mean, GenDx has a long read capabilities, although yes, you're right. That you mean long reads like long reads, or long reads like long reads. Yeah, okay. So also have to define what long reads mean. So short reads, is between 150 to 400, really. Even with the commercially available Nanopore-based HLA software, they're technically short reads. They're less than 2,000. So really, when the field talks about long reads, you're talking about thousands of bases long. And so yes, those software do use shorter fragments of DNA to get the HLA genotyping, the commercially available kit software. That being said, the all-in-one test that we showed, I hope no one from GenDx is on, but we hacked their software. So you can actually put long reads into there. You do need to do a little bit of tweaking, but really it's just an aligner, and they've optimized for sort of intermediate read, but it will take longer reads if you know how to hack the alignment.
Okay, excellent. So we have a question here. Any experience with long-read sequence using hybrid capture enrichment? Thought in implementing this strategy in clinical space?
Yeah, so that we have tried the hybrid capture with the CareDx assay. And actually, if you look up the project, the 19th International Workshop, the project that I'm a co-lead on for the ABO genotyping, that is a hybrid capture approach. It's actually a drop-in primer on the CareDx HLA genotyping assay. So I have a little bit of experience with the hybrid capture. Yes. Sorry, was the question about long read hybrid capture or short read hybrid capture?
A long read, but is actually CareDx long read? I don't know that.
No, CareDx is not long read, it's short read. Well,
Yes.
Illumina-based hybrid capture with a drop-in for ABO is also hybrid capture, but it's short read. So, but I don't know if there is a long read hybrid capture format.
Yeah, I don't know as well. The next question here is how accurate is Nanopore sequence? Because as per my understanding, it has a background noise. So leave you to answer this one.
I love that this question still comes up. So that's almost like well, it's not yet at the level of let's say take the gold standard. The gold standard being Illumina based. And there are many ways of measuring accuracy. I'm just going to take raw base accuracy that's quoted, the stats that are often quoted. So Illumina's raw base accuracy is well into the 99.5% range. Raw base accuracy with the newest chemistry, newest flow cells, the 10.4 flow cells from Nanopore is up to the 94-95% accuracy easily. So it's not as quite there, but it's getting there. If you're looking at Roche is also in the 99% range. Long read Roche is in the 99% range for raw base accuracy. And you can, with long reads and with them massively parallel sequencing, although the raw base accuracy is slightly low on Nanopore, you sort of take the cream off the top and then you get a little bit better accuracy. You just keep sequencing until you get what you want. But it will get there. I think 95, 96. If it doesn't hit 99 next year, I would be surprised. Continued improvements in chemistry and more data for their neural network training, because they require huge amounts of data for training the electrophoretic profiles. They use neural network training for that, will improve the base caller Dorado. And they released a new Dorado this year, actually. I'd have to double check what the accuracy on the newest Dorado is.
Excellent. We have another question. I think this is your favorite also. Someone asking you about the turnaround time for your long read sequence. I'm assuming they are meaning the Nanopore sequence.
So for Nanopore sequencing HLA, it's now essentially 24 hours. We could get a sample in the morning and have it out by the afternoon. Actually, we had an HSP offer on the weekend, and we came in yesterday morning, and the patient was typed by lunchtime. So that was for one deceased donor, but we do batches of you can batch at whatever batch you want, but even a batch of 12 can be done in the same day. But if it's a single, deceased donor, I think our turnaround time now is about 3 hours and 3.5 hours end to end. We report the HLA typing out by then. And that's important for us because, as I mentioned, in Q1 of 2026, we will be prospectively allocating our kidney transplant waitlist based on epitope. So we need high resolution typing of our deceased donors. So the turnaround time for our deceased donor right now is using Nanopore is less than our current time But there is an issue in that with link-seq trays, we use link-seq trays. You could set up 2 or 3 if we have really busy link-seq trays at the same time. It's not possible to do that with the workflow of the nanopore sequencing. You have to do one, wait, and then do another one, wait.
Excellent. We have another question about IT. So someone is asking, long-read sequence can generate a vast amount of data. Is this a challenge to handle in the clinical lab issue of data storage?
I must have rushed. I put a slide in IT in my deck, and I don't know where it's gone. I must have deleted it by mistake. Yes. Yeah, I should have put that. I don't know where that slide's gone, but yes, it is a huge issue with a single sample. So with our all-in-one test, one sample, the first sample we ran, the first patient sample we ran, was a terabyte of data. That is far in excess of what we need. So we've optimized for that now, but it's still huge amounts of data. To say nothing of the fact if you start look at multi-omics and integrating AI into looking at splice variants and HLA and doing all these things that we would like to take into clinic and being much more precise in how we interpret these genomic information. I actually don't have an answer. Hospital IT is so far behind what is computationally possible. It keeps me up at night. But yeah, I mean, I store my sample. I store my data on external hard drives right now. I think I have like 15 5-terabyte hard drives. That is not the way to store things. If someone drops a magnet in there, I'm pooped. But yes, big problems that everyone in healthcare. It's not just us. It's not just the HLA field. Cancer genomics is the same. Everyone is running out of space. And actually, that was what my slide was talking. It's not just us with these massive data, fields being produced for AI. Like there is no space to store data, not just for us, but for anyone in the genomic space. AWS is running out of space.
Okay. Excellent. Thanks so much, Dr. Sherwood. I think we are very running out of time, so I have to conclude the session now. There are a few questions that has not been answered, but we will email it to you, and then you can respond to these questions. So a reminder that you will receive an email with an evaluation form. Once you complete the form, you will receive your CE certificate. And we also encourage you to use the ASHU for any additional learning opportunities and upcoming webinars. Thank you again, and thank you, Dr. Sherwood.
Thanks, Ahmed. Bye.
Goodbye.